Yeah, hello again. Today, or in this video, I'd like to talk to you about the cell cycle. And the cell cycle is one of the rare things that aren't discussed very well in the ESA guide. The ESA guide is not giving enough details. From experience, you need to know more in the test than they tell you in the book. So here is a diagram of a cell cycle. So what that means is that the cell goes through a constant cycle of interphase and mitosis. So interphase is the longer phase, as you see, and interphase is the time where the cell just does its normal business and it grows and it prepares for further division. And then when it's ready to divide, it goes through mitosis. And so this part here is mitosis, and mitosis is actually only the division of the nucleus. We always say mitosis is cell division. This is slightly incorrect because mitosis are the phases of the division of the nucleus. And then this is followed by cytokinesis, which is the division of the cytoplasm and all the rest of the cell. As a result, you then get two daughter cells, which then will go back into the cell cycle. The cell cycle has 5 phases, as you saw. The first one is called G1, and G stands for growth. So this is the primary growth phase, followed by the S phase. S stands for synthesis. So that's where the cell synthesizes DNA, so DNA replicates. Then it goes through a second growth phase, and these 3 stages together are called interphase. Inter is Latin and means between, so interphase is the phase between divisions. And then is followed by mitosis and cytokinesis. The G1 stage, what happens in G1 stage? So it's the first growth stage after cell division, and this is where the cells mature. So remember, they're just divided, so they're quite small, and they have to make more cytoplasms and more organelles. And they also just carry out their normal function and their normal metabolic activities. So they might make enzymes or hormones, or if their muscle cells are contracting, whatever they're doing, they do their normal business. Then in interphase, the DNA is copied and replicated, and you know now how this works. So one DNA strand is copied, and as a result, you get 2 identical copies of that DNA strand, or not just one strand, of every single strand of DNA in that cell. So at the end of this, every chromosome is then made from 2 sister chromatids held together by the centromere. Then the cell enters the G2 phase, which is the second growth stage. That occurs then after the DNA has been replicated or copied. And now all the structures needed for division are made. For example, centrioles. Centrioles are the little organelles in the cell that later on in mitosis make the spindle fibers. And furthermore, more organelles and more proteins are synthesized, because when the cell divides, obviously it needs more mitochondria and more chloroplasts if it's a plant cell, and more endoplasmic reticulum to give a full set to each of the resulting daughter cells. So the daughter cells are then fully functional. Here is a really good link that I would like you to watch after this video. It's a website called Cells Alive. I'm pretty sure we've looked at this again at the beginning of term 1 when we started to look at cells. It's really worth exploring. They explain pretty much everything that like mitosis, meiosis, and also cell cycle. So have a look at this. And here is a revision question about the cell cycle. And now we move on to mitosis, but I leave this to you to go through it yourself.